Good morning, ladies and gentlemen. On behalf of the President of Children's Education Society and Chairman of Oxford Educational Institutions, Management, Principal, Staff and Students of the Oxford College of Engineering, I, Dr. K. Valarmati, Associate Professor, Department of Biotechnology, deem it a great pleasure and privilege in welcoming you all to the 5 days International Virtual Symposium on An Insight into Biotechnological Application. This virtual symposium brings together experts and students from science and engineering and research community and explores the technological advancements in biotechnology engineering. The Oxford College of Engineering, TOCE, is one of the most prestigious institutions run by the Children's Education Society. TOCE aims at carving competent students with extraordinary skill to face challenges of the continuous changing world. TOCE provides quality training in professional education and values in engineering, management, and computer application streams. The college has 11 research centers approved by VTU to carry out PhD and MSc engineering programs. The institution is well known for its consultancy and research and development activities. Various departments have financial assistance from reputed funding agencies, namely VGST, KSCST, VTU, DRDO NRB, DAE, BRNS, AICTE, ICMR, National Innovation Foundation, DBT BIRAC, such as and funding agencies to carry the need of young researchers and students in engineering and management.
The Oxford Educational Institutions, Bangalore offers diploma, UG and PG courses in dental, engineering, nursing, pharmacy, physiotherapy, business management, commerce, computer application, hotel management, life sciences, electronics, chemistry, mathematics, statistics, fashion and apparel technology and management, journalism, 5 years LLB, and PUC in science, arts and commerce. Contact.
With high respect, I introduce the visionary educationist, late Vidyashri S. Narasaraju Garu, the founder of Children's Education Society. In the year 1974, Shri S. Narasaraju Garu started the Oxford Group of Educational Institutions. Forever, and His blessings are there on us in all our endeavors. It is our tradition to seek the blessings of the Almighty before the start of any auspicious event. So let us start the event with invocation song.
ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ವಿನಾಯಕ ವಿದ್ಯಾದಾಯಕ ದೇವ ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು ಪಾಶಾಂಕುಶಧರ ಮೂಷಿಕ ವಾಹನ ಸರ್ಪಾಲಂಕೃತನೇ ಗಜಾನನ ಪಾಶಾಂಕುಶ वृषधरा मूषिकवाहना सर्पालङ्कृतने गजानना आदिपूजितनेनु वेदवन्दितनेनु आदिपूजितनेनु वेदवन्दितनेनु मोदकप्रियनेनु गणाधिप मोदकप्रियनेनु गणाधिप ಶರಣು ನಿನಗೆ ಶರಣು ವಿನಾಯಕ ಶರಣು ನಿನಗೆ ಶರಣು.
We have an achiever and leader who is none other than Shri SNVL Narasimharaju Garu, Chairman of Oxford Educational Institutions. It is my privilege to welcome you, sir. Hearty welcome. It is my privilege to welcome Dr. Aravind A.S., Principal, the Oxford College of Engineering, and Dr. Amarnath K., Director, the Oxford Group of Institutions. I extend my heartfelt welcome to all the participants from all over India. The Department of Biotechnology Engineering is a multidisciplinary having application in multiple fields, including healthcare, agriculture, food industry, and environmental issues. It has encompassed all the fields of life and technology and is currently a multi-billion enterprise. This rapidly advancing branch was initiated in our college in the year 2003. Since then, the department is growing vertically in terms of academics, research, co-curricular and extracurricular activities, facilitating the all-round growth of students. The department is permanently affiliated to VTU and has been accredited by the National Board of Accreditation and NAC. The department has bagged ranks in both UG and PG programs. The department has excellent infrastructure involving well-maintained classrooms and highly equipped laboratories, which are designed for today's technological challenges. The department is doing outstanding research work in VTU-approved Research and Development Center. The department has established unique research facilities, namely Center for Natural Product Research, with financial assistance from VTU, AICTE New Delhi, and DAE BRNS DRDO New Delhi. And then Center for Computational Medicinal Chemistry in Drug Discovery with financial assistance from Vision Group of Science and Technology, Government of Karnataka. And Center for Bioremediation with financial assistance from Naval Research Board DRDO, New Delhi and DBT Bharat, New Delhi. In these R&D centers, several funded research projects have already been conducted and continuing research grants have been received from various national and state government funding agencies like DBT, BIRAC, DRDO, NRB, DAE, BRNS, AICTE, VGST, KFS, VTU, KSCST, and National Innovation Foundation, etc. These projects are instrumental in strengthening our core research activities, which are beneficial to both students and staff. They have also generated sufficient amount of funds to equip our department with necessary instrumentation and other requirements. At present, the department is working on the development of bioremediation kit for the remediation of oil spill and degradation of toxic hydrocarbons under the financial assistance of DBT Bharat under PACE scheme under the guidance of Dr. B.K. Manjunath, Dean Research and Professor and Head, the Department of Biotechnology. Department is also working on developing polyherbal formulations to reduce the toxic effect of radiation during the radiotherapy under the financial assistance of DAE BRNS, New Delhi under the guidance of Dr. B.K. Manjunath, Dean Research and Professor and Head, Department of Biotechnology. The department is actively engaged in extending consultancy services to validate ethnomedicinal claims of traditional medicinal plants under the financial assistance of National Innovation Foundation under the guidance of Dr. B.K. Manjunath, Dean Research and Professor and Head, Department of Biotechnology. Highly qualified and seasoned faculties who are capable of nurturing the overall growth of students are also actively involved in conducting various programs and activities in the department. The department is proud to state that a domestic patent is filed and a bioremediation product, PetroSAP, is developed to address the bioremediation of hydrocarbons and oil spill with financial assistance under BIRAC. Now, I request Dr. B.K. Manjunath, Professor and Head, Department of Biotechnology, to give an overview about the objectives of the International Virtual Symposium on an insight into biotechnological application. I welcome you, sir.
Okay. A warm welcome and very good morning to one and all present here. It gives me immense pleasure in welcoming all of you to this 5 days virtual symposium. In the dynamic and competitive environment, it is very challenging for the Engineering Education Institute to prepare future leaders and engineers. To follow the rhythm of extremely fast and globally developing India, the 21st century engineers should have the capability to deal with the global issues and addressing sophisticated multidisciplinary and multicultural problems. They need to develop their minds to think in multidisciplinary approach, and they should be trained in a way to link topics of different subjects and disciplines in order to propose the most suitable solutions wherever society demands, especially in the pandemic situation like this. Hence, the educational institutes are not just constrained to knowledge transfer from teachers to the students, but are also involved in knowledge assimilation, knowledge generation, and knowledge spreading. The main objective of this 5 days virtual technical symposium is to bring experts, researchers, and innovators from academia, R&D institutes, and industry in the different fields of engineering and science subjects together, and to provide them a forum for knowing what is happening in the research arena, identify and conceptualize new ideas, and sharing their valuable findings and thoughts. The symposium also aims to create research interest in the minds of young graduates to pursue research as their career. This symposium will facilitate and promote interdisciplinary research among students and helps in reducing the gap between the different engineering domains. Hope this effort will help us in understanding the various domains of biotechnology, and I extremely happy to convey my heartily felt thanks to all the participants for joining hands with us. Come, let's make Atmanirbhar Bharat together. Thank you.
At the outset, I welcome today's our guest of honor and our alumni, Mr. Mukunda S.K., Senior Engineer, Arcturus Therapeutics, San Diego, California, USA. He has worked as a process engineer with 8+
Good
years
morning,
of
ladies and
experience
gentlemen.
in
On behalf of the President of
CGMP
Children
fermentation biologics like pDNA, fungal mRNA, drug substance manufacturing. He has an undergraduate degree in biotechnology and has a master in business and science with a focus on bioprocessing and business of bioscience from Keck Graduate Institute in Claremont, California. Also, he is certified in production and inventory management from APICS. Involved in fermentation and cell culture operations, chromatography, TFF purification in process and final filtration, simulation studies, process optimization, technology transfer, filling operation, cell banks and cryopreservation process, production and inventory management. On behalf of the management, staff, students of the Department of Biotechnology, the Oxford College of Engineering, we would like to extend a very warm welcome to Mr. Mukunda. Welcome Mukunda, Mr. Mukunda. Hello.
Hello.
Yeah, Mukunda, now you can share your screen. Welcome Mukunda, and you can share your screen.
Okay. At the outset, I'd like to say thanks to Dr. Manjuna, Dr. Valamurthy, and also Divakar sir for giving me an opportunity to attend the International Symposium. It was really a pleasure to listen to some of the talks by Dr. Kadir, Gurumurthy Hegde, and then also Joy Hoskeri. Unfortunately, I was not able to listen to the talk by Mr. Joshua because it was a little bit late in the night for me. So it's indeed a pleasure to talk to you young minds who are getting a good education from the Oxford College of Engineering. I remember my days over there about 14 years ago or so, and we got really good exposure to some of the topics over there. So this presentation, it's going to be a very, very broad overview of what you actually learn in your syllabus. I was just, browsing through your syllabus for the year 2019 or 2020. And I think a lot of the topics that I'm going to touch upon today it's already present in your syllabus. It's probably being covered in a lot more detail than what I'm going to touch upon today. But the interesting thing is this is not research. I am not into research, and this is more into production, operations, and those kind of things. So it may be a little bit unorthodox for you folks to get this kind of information from a research kind of symposium. So I was being introduced, I am a process engineer, nothing fancy. It's something that we learn over the 4 years over there. A lot of theoretical concepts are being taught to you in the engineering program. And then later on, I was able to join a program where there was a lot of focus on both business concepts as well as, technical concepts in bioprocessing. So that kind of molded my career to work in interdisciplinary regimes, where both technical aspects and business aspects are involved. A little bit is that I like to travel. Been to a few places, 4 or 5 countries. And then one of the things that I like to do in my spare time is to try vegetarian dishes and desserts, in a lot of different cuisines. The other thing that I'm really enjoying these days is spending time and learning a few things with my daughter. She's really young, about 2 years now. So a lot of challenges come with that. And then it's an interesting phase in my life. So stepping ahead. During the symposium, you've had talks from India, Malaysia, Australia, even in the US, there was a speaker from Boston. And this concept, whatever we're going to discuss today, it's mostly going to come from San Diego and my experience in smaller companies like Arcturus and At this point, I just wanted to give a quick disclaimer that most of the topics that I'm going to talk today are my own views. It has got nothing to do with either my work place or any of my previous workplaces. Just giving that disclaimer out there because this is going to be recorded and probably streamed at a later point in time. So want to be clear on that, that these views are my personal views and it has got nothing to do with my workplaces. Okay, so what are some of the topics that we're going to touch upon today? It's going to be like plasmid sequence, designing of the plasmids, some of the cell line selections and how that's going to happen. That's probably one of the areas that I've not really worked too much in detail, but I kind of it's like the starting point of what comes into my hands when I'm doing the tech transfer and manufacturing and operations kind of thing. So we're just going to briefly touch upon that. I pulled in information from different places to give you an idea of what happens in that particular phase. The next aspect is cell banking, which is the core aspect of creating the cells that are going to be responsible for all the other activities that are downstream of that. Then it's going to be a little bit about the seed and final fermentation. Then we're going to understand how you're going to harvest the broth that has been fermented and how you're going to get some of the plasmid out of the cells, the bacterial cells to be more specific. And then once you've got these plasmids out, how you're going to purify that and separate it from all the other components in a bacterial cell. And finally, we're going to talk about how the plasmid is going to be filled into bottles and stored. So at any point, if somebody has any questions, please share it with the faculty, and then probably we'll get it answered at the very end. If you do want to pause in between, that is also fine, and we can get those questions clarified. I can confidently say that I do not know the answer to most of your questions, but I'll share whatever little information that I have with the emphasis that I'm still learning about a lot of these things myself. Okay, so this image, a lot of you have come across this. It's straight up from a bioprocessing textbook by Paul and Doran. Trust me, if you choose fermentation or even plasmid manufacturing or CHO manufacturing, any of those things, you will come back to this image quite a few times in your career, because this is one basic picture that talks about all the different unit operations that's being carried out on a day-to-day basis. So what exactly is going on over here is, let's do this. You're getting microorganisms like E. coli, and you're putting in it a plasmid. That's what we're going to, that's the most interesting piece of this whole process. You want to have multiple copies of this plasmid. That's the goal of the whole production. And then you're going to put it into recombinant, you're going to create a recombinant plasmid, and then you're inserting it into the E. coli. And then this is where you're multiplying it. You're doing a small scale flask culture. You're going to put it into a mini bioreactor, mostly a benchtop bioreactors, 5 liters to 10 liters kind of scale. And then you'll move on to a little bit more larger bioreactors, the pilot scale is usually around 500 liters. And then the industrial scale it can be all the way from 10,000 liters, 2,000 liters, 10,000 to 20,000 kind of liters. And then you'll kind of do product recovery from this, whatever has been fermented, and then you're packaging and marketing. Okay, so in order to do this whole thing, what's the basic requirement, right? So you need a plasmid sequence. To explain in this current situation, we're all dealing with COVID-19, right? what is it that the vaccine needs in terms of like proteins that will like give us more immunity towards like or produce antibodies in our body to be able to like fight against COVID. You need to have like certain sequences, and that's what is the basic part of the plasmid sequence. So you can talk about like the S1 region, the S2 region, or the spike protein. You might have probably come across these terms like from articles or other sources of information in the media, like social media or even like print media. Then once you have that sequence, like how do you design that and how do you put that into like a cell line? This is where you're like sort of engineering the whole plasmid. Okay, so what are some of the basic components of a bacterial plasmid? Like what is needed in order to like have multiple copies and be able to produce like large quantities of our gene of interest, okay? So you're going to have an origin of replication. That is to be able to like, increase the copy number, and then you'll have an antibiotic resistance gene. This is basically like a selection kind of criteria. And you'll have a promoter, and you'll have restriction sites to be able to like identify whether your sequence, gene of interest, the insert is being propagated multiple times when you're like bacteria is like replicating in the fermenter. Some of the examples of like bacterial plasmids are pucks, PBr322s, PETs, PR6K, and PGEM. Again, I'm just introducing these terms. Do Google these terms. You'll get a lot more information than what you'll get from like an hour's worth of talk over here. This slide is just to like give a little bit more in-depth detail about what these different components of the plasmid are. It's taken straight from the source over here. I'd highly encourage people to like go out and look at this link and get more information. I found this source to be like pretty useful, and it seems to cover a lot of these topics in comprehensively. It's a really busy slide. We don't need to worry about this for now. During your free time, please free to go to these websites and get more information. So what are the different types of plasmids that are around there? It could be like a cloning plasmid, an expression plasmid, gene knockdown plasmids where you're like trying to understand what's not there, and then you produce things. Genome engineering plasmids, reporter plasmid, viral plasmid. These are the different types that are out there. I mostly deal with like expression plasmids, where we are producing like mRNAs or peptides or small sequences of proteins that will be used as part of vaccines. Again, a lot of the information about these different types of plasmids can be found in this website that I shared previously, and I'm sharing it again. You can look at the definitions of a lot of these different types of plasmids and what they're used for. More information in the day and age we are right now in, like a lot of the information can be found online, and it's a good practice to go there and get to know a lot of these things on a regular basis, because technology is probably moving way quicker than you can keep up with it. So I encourage students to like go ahead and learn things online. Okay, moving further, like what are some of the antibiotic resistance genes that I mentioned in a couple of slides? Kanamycin, chloramphenicol, tetracycline, ampicillin, and streptomycin. The reason we use these antibiotic markers is because you want to be able to grow only those bacterial cells which have these plasmids and basically kill all the other competing bacteria that's in the media. So as long as you have these resistance markers, the cells will propagate. The other cells, because they don't have that, are going to be killed, and the mode of action for each of these antibiotics are listed over here. So for kanamycin, it basically binds to your 30S ribosomal subunit and it kills that. With chloramphenicol, it's 50S. Tetracycline is very similar to kanamycin. With ampicillin, you're basically like killing the cell wall, like the cell wall synthesis is inhibited. With streptomycin, a lot of the protein synthesis is inhibited in the initiation phase. And how much of this antibiotics do you use generally in your media? It depends on where you're working. A lot of research papers will give you some information, but in general, it's going to be 50 to 100 micrograms per mil of the culture. It's not going to take a lot because the bacterial cells are like so tiny. Okay, so once you have had this antibiotic resistance and you have this like inserted gene and everything, how do you confirm if the plasmid that you've designed and cloned into the E. coli is working properly or not? There is like a few different kinds of tests that you can do. One of the most basic and the simplest thing is to grow the bacteria. Like if you grow the bacteria in the media and it grows, fantastic. And if there is nothing else that is like competing with your cell line of interest, then that's what you want. You have achieved your goal for this particular phase of plasmid construction. The other way you can confirm the plasmid construction is by doing sequencing. A couple of types of sequencing is like Sanger sequencing and next generation sequencing, where you check bidirectionally the plasmid sequence and make sure that the sequence of protein or whatever that gene that you have inserted is correctly propagating without any mutations or other errors that you generally observe. And like I mentioned, the last thing is to be able to make sure that there's no fungal and bacterial culture contamination. You want only your bacteria of interest to be able to grow and nothing else should be growing, and you need to keep that culture pure. That's one of the challenges, and it's tricky, but it's also feasible and you'll be able to achieve it. What are some of the operational challenges in plasmid design and growth? This is where, I kind of distinguish myself from the bench scientists. The operational challenges are some of the things that a lot of scientists don't really worry about a lot because they're busy dealing with 10,000 designs and so for a program. When they're finally able to zero in to 100 designs or a 10 design kind of level, that's where we kind of engineers step in and we take up and be able to expand it to and scale up the process. So when we get these constructs from the pen scientists, we ask them oh, is it growing at 25C? Is it growing at 37C? Where do you see its best growth kind of thing? And they'll be oh, we're not really sure. We left it on the bench for over the weekend kind of thing, and it grew, and this is how we were able to get. Those kind of things is what you generally hear from bench scientists. And then we are look, that's not really feasible when you go into scaling up. like we need to determine like one particular temperature, and then we should be able to grow it. So we do like a lot of design of experiments to determine what is a good temperature for growth, what is a good temperature for inducing the plasmids and such. And here, these are some of the challenges that you'll come across when you're dealing with as engineers. The other aspect is like transformation issue. A lot of the times, even though you're having your gene of interest in the electroporation components, it may not be taken up by the cell. And that is where you know that your gene of interest or the insert has not even been fused with the other components of the plasmid. So you have to go back and look whether, the sequence is good, was there any problems with the cell line, and you have to hash out those kind of topics. And then the next thing is like copy number suitability. Some of the sequences may not work in a high copy number plasmid. When you're trying to replicate like 1,000 or 500 copies per cell, it does not work. Like, for example, let's say your sequence is like 20 kilobase pairs. It's probably too huge for the E. coli to be able to like replicate itself and at the same time be able to make more plasmids. So you'll have to look for like a different cell line. Like, for example, the cell lines that I generally work with is like DH10Bs, DH5 alphas, NEB stables, or EP300s, those kind of cell lines. We work from small constructs all the way to like extremely large constructs like 20 kb, 22 kb kind of constructs. So it depends on which cell line is suitable for which kind of sequence. The choice of antibiotic also makes a difference. Like some cells are happy with kanamycin. Some other cells, you need to substitute kanamycin with like ampicillin or streptomycin. So you kind of need to play around those kind of aspects. The next thing that influences a lot of the sequence with plasmid is the content of G and C base pairs. If you have a very rich G and C content, then most of the times what happens is your plasmid is not propagating or it's not replicating sufficiently, and you do not get like good expression either. So that's where you go back and you'll be like, hey, can you please work on creating a different sequence maybe with some like less GC content? And then you talk to your scientist and then they'll work on it and they'll come back with like a different sequence. You'll be able to maybe make it work the next time. So some of the other challenges that you face is to be able to retain repeat sequences and also like retaining a really long poly-A tail. So I recommend a lot of you students to like go ahead and look at what poly-A tail is, because messenger RNA vaccines are like a big thing these days. We are working on one. Hopefully we'll be able to get something out for COVID in the next, quarter or half a year or so, and we'll be able to like, administer to the folks out there. So retention of the repeat sequences and poly-A tail, like these are some of the things that also pose a challenge, because a lot of the times, for example, with poly-A, you have like 100 to 150 kind of continuous base pairs of just adenosine. So the bacteria should be able to like keep replicating that sequence over time, and it should be stable. So that's a challenge for the bacteria, and you got to like kind of go and do some tricks with it to be able to retain the poly-A tail. So that was the spiel about all the plasmid design and some of the topics that you folks read about in different semesters with regard to the plasmid design or these things. I couldn't exactly pinpoint which semester it is, but I think once you have this kind of information, you can go back and always like, if you're interested in these kind of topics, you can focus more on those modules when you're reading in your semesters. A lot of these topics are like in the earlier semesters, and students in like the 7th and 8th kind of semester, you folks have already gone through this. So you're probably aware of these topics. How's it going so far? Good? Maybe somebody in the faculty can say how's it going? The pace okay?
Yeah, it is fine. It is fine, Mukunda. You can continue.
Okay, thanks. So these are the other topics that are relevant for Plasmid design. And I can share this presentation with the faculty, and then you folks can, if you want to come back to any of these slides, then feel free to come back and be able to like, gain more information or gather more information. Okay, so moving on, we're going to step into cell banking. What are some of the challenges and interesting things in cell banking? In a very basic overview, what you're doing is you want to be able to provide adequate supply of well-characterized cells that are going to be equivalent whenever you take it and try to produce your plasmid. That's the basic goal of cell banking. And you're going to make enough cells that will last over the lifetime of the product. So basically, you want to make enough cell banks that's going to last for like about 5 to 10 years over the life cycle of the initial phase of a product. And then in between, you can do like another bank and then be able to like compare and contrast and all that. There are 3 stages in cell banking. The first stage is what comes from the plasmid design and growth. From that, you'll get about like, 10 vials of banks, like 10 vials. When I say that, it's about like 10 mils of culture. And that is used to create your master cell bank. The 10 mils is your research cell bank. And then from that, you create a master cell bank. And from the master cell bank, you create what's called as a manufacturer's working cell bank, or MWCB or WCB. The terminology varies in different institutions wherever you're going to go work. So this is the basic overview of cell banking. So here I wanted to kind of like, introduce you to like some more pictures, because it helps to like visualize things a lot better. Here we have the electroporator, and there's like an electrical schematic that you folks, whoever is interested, can always like go look at what is being done and how it's used. One of the important topics for cell banking is to learn and to understand and use this particular thing right here. It's called an inoculation loop. It might look very simple, but I emphasize on this fact: do ask your faculty and do ask anybody else to learn a good technique of using this. If you ever go into like this area of work, you will be appreciative of the fact that you learned how to use an inoculation loop very early on in your career. So you're going to use this to basically like streak the plates and to get these isolated colonies. Again, the emphasis is on isolated colonies. So whenever you're having practicals, lab, go ahead, learn to use this properly, and then you'll be very thankful later on in your career when you're able to like produce these kind of plates where you can like pick isolated colonies. This here is a pretty standard incubator. This is an incubator shaker. Same thing, an incubator shaker where you kind of like pick the colonies, then put it in this liquid culture and you let it grow. And that's how you multiply the bacteria and so on. Okay, so now that you know the basics of what is happening, this is a process of producing cell banks. So we kind of start with like competent cells. These are the cells that are like DH10Bs, DH5 alphas, New England Biolabs stable cell line, or EP300s, or other kind of cells. Along with this competent cells, you add your plasmid DNA sequence that you've designed, and you add about 1 mil to like 2 mils of media, and then you electroporate it, and you have your like basic cells, and you incubate it at, let's say, 24 degrees Celsius, or 30 or 34 or 37. A lot of those things, a lot of that information you're going to get from papers. You don't need to worry about that too much, or you'll have like some senior faculty or senior folks, scientists at your company that are going to like kind of help you with that. Unless you're doing like a startup, even in that case, you're going to mostly rely upon papers for this kind of information. So you're going to grow it at a certain temperature, at a certain RPM, for a certain time, to be able to like multiply the bacterial cells. Usually it's like overnight, you do it for like 12 to 18 hours, and you do it at like 250 RPMs to about 330 RPMs kind of thing, and you grow the cells. So once you've done that, you're going to dilute either 1 to 10 or 1 to 100 of those cells, and then you're going to streak the plate. This is kind of like a key operation. Like I mentioned in the previous slide, you want to be able to isolate that colonies that are like well isolated. You want to be able to like pick colonies that are well isolated. So you streak 3 to 6 plates, and then you identify well isolated colonies, and then you again incubate or inoculate test tubes. So what exactly is happening here is you're going from like solid media to liquid media. You want to be able to test the colonies that you have picked are able to survive both on solid media and liquid media. And then once that has occurred, you go back and you streak a couple of plates, again, basically identifying 3 to 6 well isolated colonies. And finally, you're going to inoculate the shaker flasks, which have about 50 to 500 mils, which will become your actual cell bank. You're going to incubate them overnight, and finally, you're going to add glycerin or any other cryopreservative, and you're going to mix well to get like a homogeneous mixture, and you're going to aliquot into like 100 vials or 300 vials. Usually for master cell banks, it's going to be like between 100 and 300 vials, and you're going to use one of those master cell bank vials to create your working cell banks, and that's what you'll use on a regular day-to-day basis. And the master cell bank, you're going to treat that as your family's jewels, like your grandfather's or grandmother's jewels kind of thing. You're going to like preserve that and save that and protect it with your utmost care, because that needs to be used for the next 5 to 10 years. What are some of the quality control aspects that you need to do when you're doing cell banking? You need to check for the host cell identity, whether it's gram-positive bacteria, gram-negative bacteria. Usually it's gram-negatives with E. coli. And then you're going to check for the bacterial colony morphology, whether all the colonies that you've grown in a plate are similar. They have like the same spherical structure or ovoid structure, whether they all appear yellow, white, creamish, depends on the cell line and the culture. you're going to test for DNA homogeneity. You're going to make sure that the plasmid is the same across all. You're going to pick a couple of colonies and you're going to test that. The antibiotic resistance, whether it's being propagated. You're going to check for the plasmid copy number, whether high copy number is staying high copy number, or if it's going to go into a mid copy or a low copy. You're going to check that. You're going to do some restriction digest analysis on agarose gel. And then you're going to check for the host cell purity. You want to make sure that there is no contaminating fungus or other strains of bacteria that is growing along with your cell. You're going to check for lytic phage contamination and lysogenic bacteriophage contamination. You do not want any of these phages in your culture because they're going to kill your bacteria and you're not going to get any plasmid after that. And you're again going to confirm your plasmid identity by doing sequencing. And finally, I mean, this list is not comprehensive, but these are some of the tests that in general give you like good information about how your cell bank is done and whether it is well characterized or not. A lot of these things are required for regulatory submissions to either the FDA or the EMA or even Japan or other regulatory bodies. So it is important to do these tests and to show that there are no issues with your cell bank. So what are the challenges in cell banking? You need to be able to zero in on the growth temperature, and you definitely want to make sure that your growth is, your cell banks are sterile. There is no fungal contamination. There is no bacterial strain contamination and such. Then I can't emphasize enough on learning a good streaking technique and to be able to like choose an isolated colony. It's important to pick an isolated colony because you don't want to have two different types of, or two or three different types of bacterial colonies in a culture. You want a uniform and homogeneous culture. And you need to be able to like define the parameters and to be able to like justify your choice when you're selecting a colony. You want to be able to like say, look, this colony is able to grow well in the next 12 to 18 hours, or this colony produces a high copy number of the plasmid that we need, or this colony is giving good results with like density, like it's able to like replicate and produce kind of thing. So those are some of the choices that you make in your company or in your research kind of thing. There'll be a lot of guidance from other folks that will help you. The goal is to be able to like convince why you're picking such a colony and what are the benefits of that particular colony. And lastly, some of the challenges that you face in cell banking is to be able to utilize the space inside a BSC or a laminar airflow. Probably in your labs, faculty are teaching you how to do this. And I kind of like want to share that like when I was at Oxford, I was taught a really good technique, and it's kind of like stayed with me over the next 15 years now. So it's important to like learn what's a dirty area, what's a clean area, where you're going to keep all of your materials inside the BSC, how to keep your hands inside the BSC. You should not be resting your hands on the plate out there. Like the air patterns are in a certain way, like you should not disturb the air patterns, be gently moving inside the BSC, those kinds of things. Like a lot of those things you learn in practice, do pay attention to that whenever you're taught, and it'll help you a lot later on in your life. So these are some of the topics that are relevant for your cell banking kind of processes. So the next topic is fermentation. what's the big picture in fermentation? You're making media, you're doing a small seed fermentation, and you're doing a final fermentation such that you've produced enough cells. So the goal is to provide a suitable environment and all the nutrients for the growth of bacterial cells and for plasmid production. The goal again is to like produce enough quantity of biomass and the desired quality of plasmid. There are multiple ways of getting to these goals. You can do like a batch fermentation, you can do a fed batch fermentation, or you can do like a perfusion system whenever you're dealing with like mammalian systems, CHO, humans, and other organisms. So this is like a really big picture. In order to get to the big picture, you use these things on a day-to-day basis. Flasks. You have used this. There's like baffled flasks. There's like plain bottom flasks. Some of the flask caps are open. Some of them like have a membrane to be able to like allow for oxygenation and such. Some of them are closed containers. These are pH probes. You need them to be able to like monitor and adjust the pH as necessary. Bacteria generally is happy around like 6.5 to like 7.2-ish kind of pH. If you go either low or high, you're going to like add ammonia or like supplement with like other nutrients to be able to like bring it back into the happy zone. And this is a dissolved oxygen probe, DO probes. You measure how much oxygen is there inside your culture. It's kind of key that you maintain the oxygen at a certain percentage. 30% or 50% is generally like an accepted percentage for bacterial growth. Probably seen these benchtop fermenters. It has like an exhaust condenser, an inlet filter, pH probe, DO probe. You have like an impeller, couple of impellers, the agitator, and such. And one of the challenges that you face in day-to-day, like if you deal with these benchtop fermenters, is that it's the assembly of these head plates. They're kind of tiny, and if you have like big hands, kind of like mine, like you're kind of like struggling to like open one of these knobs or like put this tubing in there. So, day-to-day, that's one of the challenges that I kind of face when I'm working in process development and I'm like dealing with benchtop fermenters. With larger stainless steel fermenters, like it's a little bit easier where you have like things are like spaced out a lot more and it's easy to work with. Again, this is just a different brand of the fermenter that's around. So this is like a single-use fermenter. This is like a 300-liter fermenter, I think, and this is a 30-liter fermenter. You use Delta V controller software to like basically control the different aspects of a fermenter. And this is another area that if you want to go into, it's really a good challenge to like deal with like electronics and be able to like communicate with like the instruments over here. So that's something that, if you choose as a career path, like it's very interesting, it's very challenging, and, a lot of satisfaction when you solve problems around the communication and dealing with things. We also use like density measurements. This is a nanodrop spectrophotometer. Basically using this day in, day out to like measure the density of the bacterial cultures. You do measure the density at 600 nanometers. You probably are aware of that already, and you do that in your lab. Okay. So now that you have like some of the images in your head, like we can step into the fermentation process. So the first aspect of this whole chain of events is to be able to prepare media. A lot of the basic components is to provide like a nitrogen source, a carbon source, and oxygen and such. But in terms of the media, you're basically like providing nitrogen and carbon source for the bacteria to be able to like produce, replicate, and to propagate the plasmids. Some of the time is like invested into like setting up your fermenters, your mixing tanks and such, and also like BSC. You need to like move all the things into the BSC, like wipe it, clean it, and those kind of things. It's usually like a day and a half kind of amount of work to be able to like do these things. Once you've done that, you need to perform calibration on your pH and DO probes. It usually takes about like 20-30 minutes to do this, but it's a very critical aspect of your process, and you want to do this correctly because there is a lot of like feedback interaction from this measurement into the whole process. So calibration of the pH and DO probes are important. So once you've done that, you move into like thawing one of your vials. If it's like a research lab, you're generally just working with research cell banks. But if it's like more a GMP process, you deal with like working cell banks or like a master cell bank. You want to thaw the vial in a controlled manner. You can use like a heat block. If not, then you can just hand warm it in your hand and then use that. So once you thaw the vial, you're going to inoculate your seed fermenter. It's generally like a smaller fermenter, like the one in the previous slide. You can inoculate like a 50-liter fermenter or a 30-liter fermenter, and then you're going to incubate the cells at like X degrees C for Y time until your density is going to be like Z at like 600 nanometers. So usually you want to have a constant density of the cells that's going to go into your final fermentation because you want to have a controlled process. You calculate the seed density, and once you have achieved the enough number of cells, you're going to transfer that amount of inoculum into the fermenter. And this is where in the final fermentation process, you're going to monitor your pH, your dissolved oxygen, your temperature, and there's something called cascade. So as I was mentioning previously, the dissolved oxygen is going to need to be maintained at either 30% or 50%. And the reason you want to be able to do that is to provide enough oxygen to the cells to be able to grow. And as the cells are multiplying in the fermenter, the oxygen consumption is going to keep on increasing. That's why it's going to drop from like 100% all the way down to like 30%. And when that happens, how can you make sure that you're providing enough oxygen? So you can increase your agitation, or you can supply more air or more oxygen. And cascade is a term that's used to be able to get the feedback information from the dissolved oxygen probe. The reading is taken and translated back to the controller unit where it says, oh, pump more oxygen or pump more air, or increase the agitation rate. So you need to turn that on when you're setting up your final fermenter. And once that's done, you can do, in the simplest of terms, you'll do a batch process where you have provided all the nutrients and the media and everything to the cells in one go before you add the inoculum. In a fed batch process, you're going to provide that, and later on you're going to add a little bit more of your concentrated media where it has all the supplements and everything. And in a perfusion process, you're constantly removing your spent media and you're giving it fresh media so for the cells to like be able to grow. Batch process, it's the simplest of all. You just provide everything, you're done, and you just let the cells grow. End of the day, you go in and you harvest the cells. So once it's been growing at X degrees Celsius, in order to like increase the plasmid production, you shift the temperature from, let's say, 30 degrees Celsius to 37 degrees Celsius, or 37 degrees Celsius to like 42 degrees Celsius. And what happens when you do that is the bacteria will start producing plasmid and stop replicating. So initially the goal is to be able to like replicate the bacteria, and then after a certain duration to shift the temperature and make the bacteria to focus on producing the plasmid. So this in general is the fermentation process, and I absolutely love this process. I've spent a lot of time, close to 5 to 6 years in this process, and the challenges are quite good, and it's again like really satisfying to like solve problems and to be able to like get products out there into the world. What are some of the quality control aspects that you need to do during fermentation? A lot of this is pretty similar to what you do with cell banks, because at this stage, all you're doing again is increasing the number of bacterial cells and being able to like, produce enough plasmid. So the tests are very similar. You're doing like a host cell identity, checking for morphology, checking for copy number, you're doing restriction digest. That's a key thing at this unit operation stage, because bacterial cells like have gone through like so many generations at this point in time that you want to make sure that the plasmid is still being retained. And then purity, I can't again emphasize enough, that's important. And you're doing like plasmid identity by sequencing, and you're also checking for viability. What are some of the challenges in fermentation? Sterility. It's very important. So whenever you're doing these inoculum transfers, media transfers, you're putting your pH probe, your DO probe, you want to make sure that it's clean. One of the ways we can get around this is to do an activity called steam in place, where once you have set up your entire fermenter, you're going to put in steam and you're basically like sterilizing your media for like 30 minutes or 45 minutes or such. And then that way that the inside of the fermenter is completely sterile. This is how you do with like stainless steel equipment. If you're using single-use fermenters, which is getting more and more common these days, you make sure that all the transfers occur sterile, and you have your pH and DO probes. They're already inserted in there from when the bags come from manufacturers, and make sure they're operational. BSE techniques, again, you need to be able to make these sterile transfers, so you need to learn how to use the BSE. Some of the challenges involve around setting up of the equipment. The pH probe, the DO probes, the agitation, the cascades. Whenever there's like a new cell line, you need to be able to understand what conditions are good for it and how you can help it to grow fast. Some of the efficiency goals that you may come across are like, why do the cells need to grow for like 20 hours? Like, why can't you make it grow, in like 15 hours? 5 hours savings translates to a lot of like, money you're saving on like people, you're saving on like power, you're saving on like other things. So efficiency goals kind of drive how you run the process sometimes. And the other aspect is to be able to consistently provide the same yield and quality over time. So whenever you're producing a product for like, let's say 5 to 10 years, you need to be able to like get the same quality every time. The other challenge is to like clean the equipment, which is why a lot of the processes these days, they are moving towards being single use. With single use, all you do is like set up things, and after your process is done, you can just throw all the bags, the plastic, and other things. With stainless steel equipment, you got to make sure that between batches, your equipment is clean. And you're going to use like different solutions like, sodium hydroxide or phosphoric acid or other cleaning agents to be able to like make sure that your equipment is clean and ready to be used for a different product or process, depending on what kind of industry you're in. If you're working in like a contract manufacturing organization kind of place, you're dealing with like different products. So you have to doubly make sure that you're not contaminating your X customer's product with like Y customer's product. So you need to make sure. And the way you do that is by checking for like, you take swabs and you check the interior surface and you make sure that all the ports are clean and so on and so forth. Okay, so here's some of the relevant topics for you for fermentation. I think in like 7th semester or so, you have like a whole semester's worth of modules. So you're going to learn like a lot of detailed stuff. This is one of the labs, I believe. Like I kind of find it really interesting. Do pay attention to all of these things. Like you may think that these are like really simple topics, but those are the simple topics on which you'll be making a living one day in the future. Design of fermenter. A lot of these things are pretty standard in the industry. You can learn the concepts, but there's not really much you can do to change things wherever you go to work. This is the fermentation technology or bioprocessing subject that you have in one of the semesters. So I kind of want to emphasize on this capital investment, manufacturing costs, and those kind of topics. Like for me, since I've done like an integrated program in like both business and technical side of things, I get to be involved in determining costs and to be able to like work with vendors to determine what kind of equipment we need, how much does it cost, like why does it cost this much, why can we, can you reduce it by 30%, and those kind of negotiation. So if you're interested in that kind of financial aspect of the biotechnology or pharmaceutical kind of industry, like this is a good topic to like focus. And you can probably do like an MBA or those kind of things after your engineering, and you can kind of fit in different kind of departments in the biotech and pharma industry. This again is some of the interesting topics. Like this is one of the labs where I think you're basically learning whatever I'm speaking during this talk. Like you're learning how to break down the cells and all that kind of aspects of it. You're learning how to do the chrome process and those kind of things. So when you're learning how to prep the fermenter and produce all of this, like those are some of the topics that I think if you focus upon, like it'll be useful for quite a long time when you're working in the industry. Okay, So now that we have made enough cells, we need to harvest it and lyse that to get the plasmid out. So if you consider this to be like the E. coli cell, we are only interested in this tiny little plasmid, and we need to separate that tiny little plasmid from all the other components that you see here. That's the goal of this particular unit operation. And how do we achieve that? 3 simple steps. You have like a centrifugation aspect to remove or separate the cells from this media, and then you're going to do some depth filtration after you break down the cells. And finally, you're going to do some tangential flow filtration where you're concentrating and doing some buffer exchanges. So again, the big goals: to separate cells from the spent media, to lyse the cells, to separate the plasmid. It makes somewhere around like just 3%, and the host cell components are like DNA and protein, which is about like 55% of the composition. You have like RNA at about like 21%, and there's endotoxin. Endotoxin is a really hard thing to separate, so you do like a completely different unit operation sometimes to like just get rid of endotoxin. The goal again is to just separate process and product related impurities from your plasmid. Here's a picture of like an industrial centrifuge where, you know, your feed is coming in and then it's rotating and you have like all of these solid deposits here and your media is just going out. This is like a very cursory image of how the bacterial cell paste looks like. It's usually like brown because of the media, and it's either stored in like bags or bottles, those kind of things, and they're usually stored in freezers, -20°C or -80°C kind of thing temperatures. This is an image where you can see some mixing tanks that are used in lysis. This is like a schematic, and in general, it's going to look like humongous like this. You'll have like a series of tanks that are around. This is like a depth filter. It's called a Kuno filter. And Kuno is just a brand name. 3M produces it. They go in these stainless steel kind of housings where you put it and then you pass the filters. This is a TFF or UFDF skid from Paul. It's a CM150 kit, or skid, sorry. This is more like industrial scale. And here's a skid from Repligen, I believe, or Spectrum. This is more like a process development scale, a benchtop kind of version. It's a really cool system where a lot of the things can be automated, and it also like shows trends on these things. Get a chance, always bug your faculty or other contacts to take you on an industrial trip. A lot of folks are around at companies like Biocon or AstraZeneca or Sartorius, those kind of places. So ask your faculty and other people to arrange for an industrial tour, not during these times of COVID, maybe 6 months, 8 months from now when things have settled down a little bit. Companies might be open to students. You can join professional organizations like ISPE. They generally have tours. During my time at Oxford, I think we kind of visited Sartorius and a couple other companies. We were kind of lucky to do that as a batch, I guess. So do bug your lecturers and faculty to go on a trip to one of these companies. It's always a good thing to go and see by yourself how the equipment looks. Okay, so now that you've seen some images of what equipment is used in harvest and lysis, here's the process. So you're going to use a centrifuge, take in all the culture from the fermenter, and you're going to centrifuge the broth, retain the paste, keep the cells, and you're going to discard the supernatant. And then you're going to mix the cell paste with something called resuspension buffer. It usually has like tris, EDTA, and sugar. Tris is basically used as a buffering agent. The EDTA, it like chelates the divalent ions, cations, I believe. And it also like destabilizes the cell wall. The sugar is generally used to maintain the osmotic pressure. Like you do not want the cell to like kind of break open at this stage. You kind of want it to like still preserve its shape and everything. And you don't want like all the proteinases and RNases that are present inside the cell to like kind of go and damage the plasmid. That's why you kind of want to use the sugar. And once you've mixed and suspended your cell paste in the resuspension buffer, you're going to add an equal volume of something called lysis buffer. The basic components of that buffer is sodium dodecyl sulfate and sodium hydroxide. So sodium hydroxide basically helps to break down the cell wall. And then the SDS is used to like solubilize the cell membrane. So once that happens, all the components like inside the cell are out. They come out into the open, and you want to mix these two buffers through a static mixer. You do not want to mix it very agitated because it might rupture your plasmid and genomic DNA. So you kind of want to be gentle, which is why you use something called a static mixer. And once that's done, you're going to add an equal volume of neutralizing buffer. So at this stage, the pH, because of the sodium hydroxide, is going to be around like 12-ish or 13-ish. It's not good for the plasmid, like it's almost denaturing at this point in time. And you want to like re-unheal all the strands of DNA, which is why you use potassium acetate and glacial acetic acid, I believe, to neutralize and bring down the pH back to like about 4-ish, 5-ish kind of thing. And then once all that's done, you're going to pass that mixture through the Kuhner filter, and you're going to discard all the debris that's settled in, and you're going to keep your supernatant because that's where your gene of interest or plasmid is. And you're going to add some calcium chloride, and you'll precipitate all the RNA. Most of the long RNAs are precipitated. Some of the short RNA is still left in the mixture. And then you're going to do another depth filtration. And finally, what you want to do is to do a tangential flow filtration or an ultrafiltration diafiltration process where you're reducing the volume for like, let's say X grams per liter or whatever is your loading condition onto your chrome unit operation, and you're reducing your host cell impurities even further. And you will adjust the buffer conditions. You'll move away from all of this sodium dodecyl sulfate, sodium hydroxide, tris, EDTA, calcium chloride. There's so many of these chemicals in there right now. You just want to remove everything, and you want to put your plasmid in a buffer that is suitable to be put on a chromatography column. So these are the different steps that occurs in a lysis process. So what quality control do you need to do at the end of lysis? You want to check the appearance of your plasmid. And at this stage, you also want to test if your plasmid is supercoiled, linear, open circular, or a dimer or a tetramer kind of thing. And for DNA vaccines in general, like you want just the supercoiled form. You want to separate the supercoiled form from the other forms of plasmid. And then you check the concentration of your nucleic acids. You still do not know the concentration of plasmid yet because there is a lot of other host cell impurities and RNA and such. You've not separated them completely. So at this stage, we call it just nucleic acid concentration. You check your pH, you check your conductivity, because those are the key parameters when you're loading onto an anion exchange column. And then you check by doing a restriction digest again on gel. You check for residual protein, you check for residual antibiotic, because you do not want any antibiotics left when you're injecting plasmid into people. What are some of the challenges with lysis? It's mostly to do with how you make buffers. You want to make sure that all of your buffers are made to the correct specification. Each of these buffers are generally having a specified pH, a specified conductivity, and a specified other releasing aspects. So you want to make sure that your buffers are done correctly. And once your buffers are done, you want to make sure when you're mixing your resuspension buffer with your lysis buffer or the neutralizing buffer, you want to be able to make sure that there are no zones of high or low pH. The mixing should be very homogeneous. And once you do that, you will have homogeneity. Mixing is a challenge that you kind of like overcome with experience kind of thing. And the other aspect of mixing is to be able to use pumps correctly, to be able to achieve the proper flow and the proper shear rate. So if you have any practicals where you're using pumps, learn how to use them by going through the manuals and such, like what tubing goes to them, if the tubing is like what material the tubing is, like platinum cured silicone or a different type, like a Masterflex or those kind of things. Like you need to know the different types of tubing. And setting up of the equipment, like that's going to be a standard thing with all of these unit operations. With TFF particularly, I want to emphasize that you need to understand how much material you can load per area of your filtration membrane. If you load too much, you're going to have fouling. If you load too less, you're going to need such a large amount of filtration area that you might not be able to accommodate in your space. So you need to kind of optimize the material load and the membrane fouling. And last but not the least, again, you need to consistently maintain the quality and yield. Here's a relevant topic for lysis. Emphasis here is on using of the pumps and also to understand the different types of filtration and types of equipment that's used in that. Okay, we're almost there. Purification and then fill. So what's the major goal of purification? So you're going to use chromatography for purifying. Two different steps here, anion exchange and hydrophobic interaction before you do tangential flow filtration yet again. So what's the goal under anion exchange? So under the appropriate salt conditions, the plasmid binds to the column. This is a bind and elute based chromatography. So it separates the host cell impurities like the DNA and RNA and proteins. Once the plasmid has been bound to the column, you're going to use a higher salt concentration to elute the plasmid in this particular step. With HIC, what you're doing is under the appropriate conditions, the impurities bind to the column, the plasmid flows through, and this particular type of system is basically used to reduce endotoxin. So if you can find a good system where your anion exchange is able to remove the endotoxin, you do not need to do this step. And that's the challenge that a lot of the companies are focusing on these days, to be able to like reduce or combine these two unit operations into one. And how do you do these things? Well, you have something called like an actor. Actors are pretty standard chrome systems that are used in most of the companies. This is how the actor kind of looks like. What is it basically is it has a set of pumps. This is where these are the pumps that you use for sample loading. These are the pumps that you use for mobile phase A, mobile phase B, and then you can use these kind of valves over here to kind of like mix and match your inlets. It's basically creating a flow such that it goes through the column and through like different sensors like the pH meter, the conductivity meter, and the density meter, basically a spectrophotometer, those kind of things. And then it comes out, and then you can kind of collect your product in these automated sampling systems. This is a picture of like how different scales of columns are used, different types of resins are used. If possible, when you're having practicals, learn how to use this column housing. I do not know if any of your syllabus requires the use of this, but if you can get hands-on, it's a much appreciated skill. Like even after my 10 odd years in the industry, like a lot of the deficiencies that we see, or rather like there is less number of people that know how to like pack columns and such. So this is like a skill that will be appreciated and valued if you can get it at an early stage. This is just at the PD lab. Like you also get like pre-packed columns, like somebody is making the columns for you and giving it to you. It's a little bit more expensive, but you do get that. And at the industrial scale, you also have automated packers these days. So even if you do not have that skill, it's kind of like learning how to use robotic systems to like pack the column for you. So either you can learn the skill or you can learn how to use robotic systems to like help you pack columns. This is how like an industrial scale skid looks like. It's basically the same thing, but here you can see that the tubing is kind of like bigger, and these are the different components. You can see when you get the slides, you can like go ahead and search and get a little bit more information, or you probably have already learned these from your topics in the program. An upcoming kind of Chrome system that is used these days is called monolith columns. So this is like a single column that's used for separating and purifying components. It's really kind of cool because you don't have to pack columns. Again, there are so many companies out there that do not have enough people who have that particular skill, so they just buy pre-made columns. And one of the advantages with these monolith columns is that it's just a single piece, so you do not have to do a lot of like asymmetry testing or HTTP kind of testing to qualify the columns. All you have to worry about is the residence time. And if you do not know these terms, not a big deal. You can always like, search for these terms online and you can learn these terms. Okay, so what's the process of purification using chromatography? First thing is to pack the column and qualify it. In most cases, like if you're using a monolith, you don't need to pack a column. All you need to do is just qualify it, and you qualify it based on ranges for asymmetry, which is basically the elution profile. It needs to be in a certain format within a certain range, and the math behind it is automated these days. Acta will give it to you at a click, and if you want to know the theory behind it, you can always look it up. Qualification you can do by putting in tracers like uracil or sodium chloride, those kind of things, and then you trace the profile of elution. So once you've qualified your columns, what you need to do is to equilibrate your anion exchange column and to zero the UV. During equilibration, all you're doing it is making sure that the whole column is uniformly provided with the same charge, such that when you load the product, you're able to give it good binding and be able to get good recoveries. So once you have equilibrated, you load your product, you wash the product, and then elute it. This is a very standard load, wash, elute kind of process. And here you're going to do an intermediate testing on the quality and quantity of your plasmid to make sure that you have recovered enough. Usually it's about 70% for anion exchanges. Let's say you load a gram of your plasmid, you get only about 700 mg back kind of thing. And then in order to prep your plasmid DNA for the Hick process, you add ammonium sulfate salt. Usually it's about 1 molar to about 2 molar concentration of ammonium sulfate, or if you're using salt, I think it's about 3 molar salt kind of thing. And the same process with Hick, you're equilibrating your column and you're zeroing your UV before you load the product, and you're doing the wash and dilution. But in this case, it's just going to be a flow through. So your pDNA is going through, but your impurities such as endotoxin, that's getting bound on the column. And lastly, you're going to do again an intermediate quantity and quality check to make sure that, let's say if you're loading 700 mg of plasmid, you're able to get at least 650 odd mg. Usually the recoveries is around 90% on these kind of columns. So you get that out. And then because there is ammonium sulfate in this solution, you need to do a tangential flow filtration to remove the ammonium sulfate and to put it in a more happier kind of environment where you're using either saline for injections, or it'll just be water for other kind of formulation. Basically Wi-Fi, which is water for an injection. It's a more purer water. So this is how you kind of big picture of how you do the unit operations when it comes to the downstream purification. The theory and the other aspects of it, I'm sure a lot of those topics are being covered in your syllabus, which is why I'm not going into the details of it. And there's numerous number of resins that can do anion exchange and Hick, that it's not really, you can't cover much of those topics in an hour and a half kind of information sharing. So for anion exchange, like I was mentioning, you're going to load your product, and at this point in time, you're going to zero your UV, and then you load your sample. You're going to see a good enough absorbance over there, and then you're going to wash the product that has been bound to remove residual protein and residual RNA, those kind of components, and then you'll switch your buffer, and then this is how your product's going to elute. It's going to be a nice sharp peak, And then you are going to either re-equilibrate or you're going to clean your column with like cleaning solutions like sodium hydroxide and other components like acetone or acetonitrile, depending on what you're requiring to remove. So if you can explain some of these concepts when you're like, going for your first job kind of thing, you'll be surprised at how you're going to stand out in those interactions. Here's a representative chromatogram for flow through, and you can kind of see that all of your plasmid is kind of like just coming out here, and you're going to do like cleaning process at the end of it. There is not like a product elution peak somewhere here. It's just this is where your endotoxin and other host cell impurities are being bound, and they are coming out when you're using the cleaning solution. Okay, we're really close towards the end over here. Sorry if I'm kind of going over. Quality control and testing after purification is, this is where you again like check the appearance, your supercoil. So at this stage, since we have removed a lot of the host cell DNA, RNA, and protein, you get a true concentration of your plasmid. And this is what kind of determines how much yield you've got from like your fermentation, your lysis, and those kind of unit operations. This is what kind of measures your performance for those unit operations. Check for pH. You don't want to be injecting like pH 2 into people, so you kind of want it around 7 or 7.1 or 7.2, which is like a neutral pH when you're putting material into people. You check for the identity again by gel. You're checking if there is any residual protein that's remaining, which it shouldn't be. Here it's really like trace amounts, and again, trace amounts of your antibiotic. So challenges in purification. Buffer, like that's like a big thing. When you're loading your product onto your anion exchange, you want it within a certain pH range and within a certain conductivity range. If you do not have that within tight specs, your recoveries are going to be impacted. Let's say when you're loading, you have a lower conductivity than what is normally required, and during elution you have like a higher than normal, then a lot of the thing is going to elute much before than when you want to elute. So the separation between your impurities and your product is not going to be sufficient for a good separation. So those are some of the things that you learn for different products, what kind of buffers are needed. Part of that, like I mentioned, is pH and conductivity. Learning to make these buffers, I think, is important and key. And since you're still in the stage of learning those things, I request that you learn to do those things properly. It'll kind of help you all in the longer run. Packing columns and qualification, like the asymmetry and HTTP, it's kind of phasing out. Like if you're still using some resins where you need to pack, you got to use this. But if you're using something like monolith columns, like you don't need to worry about these. Flow rates of various phases, right? So the equilibration, the sample loading, and all that. Like to achieve the optimized residence time, you need to play around with flow rates of these different phases. Like, do you want to have the same flow rate during your elution phase as your sample loading, or do you want to have it slower? This is something that you kind of like learn with different projects and different products. You tend to do like design of experiments to determine what flow rate is good, in which phase, and those kind of things. And lastly, to be able to learn how to write chromatography methods in Unicon. That's a really easy thing to do these days. You can like drag and drop different components and do it. But that programming skill is key. So if you get a chance to learn that, do learn that. And lastly, cleaning of the columns. A lot of these resins are kind of sensitive. So if you use harsher cleaning solutions, they tend to impact the ligand that is on the surface of these resins, and in turn, they tend to impact the binding capacity of these resins. So learning and using the correct cleaning solutions is an important part of the purification process. Some of the topics that are relevant, I think like these topics will go into much more detail than what I've spoken to you today. So if you're more interested in downstream kind of processing, give more importance to these topics. And fermentation and downstream, they're like two different sides of the coin. Like sometimes what happens is if you get into one of these fields, like you tend to go deeper and deeper in just one of the topics or one of the unit operations. I've been kind of like fortunate to work in smaller companies that I get to like interact with all of these unit operations, both upstream and downstream. So if you want to learn more, join a startup. If you want to learn broad, I would say join a startup or like a smaller company. But if you want to learn more deep in just one kind of area, it's kind of useful like if you join a bigger company, because you tend to like focus more on one area rather than multiple areas. Okay, we've got about just 5 more slides to go, I believe so. Drug substance fill, this is like the last part of it. The goal with this particular unit operation is to adjust the concentration of your final plasmid. You do depth filtration with a 0.2 micron filter to remove any bioburden, which shouldn't be there to begin with because you've gone through like so many unit operations and you've removed all the cells and other things. And finally, to fill, these vials like with X amount of volume and store them at, different temperatures, like either -20, -80, or in a fridge, say 2 degrees to 8 degrees, or at room temperature. Room temperature is generally not recommended unless you're like dosing patients in the hospital, but otherwise, those are the temperatures that you kind of store. To formulate the pDNA, either, saline or water or glucose solution or whatever is the correct method for dosing and at the correct concentration. To filter at an appropriate flow rate and eliminate the bioburden, and to store in the correct container and the right temperature. That's the goal of this particular unit operation. So here are some of the equipment that's used. So filters, you've come across these probably. These are just standard 0.2 micron filters. And you use these kind of bottles to store your drug substance. Usually, I think they're used PETG bottles or HDPE kind of bottles. That's the material. And you're going to store them in freezers. These are some of the industrial freezers that are out there. Okay, so this is the final quality control and testing that you're doing. Looks like I've just kind of repeated this information over here, probably not updated the slides, so apologies on that. But in general, you do a more robust testing of all the testing that you've done in the past unit operations, and you test it over here. Sorry again, like I've not done this slide. So you're going to test for your concentration, your supercoil, in general your antibiotics. So it's mostly a repetition of tests. But the main thing over here is you're testing for bioburden. What is that? Is like you do not want to see any growth of organisms at this stage because you've passed it through a sterile filter and you've bottled it. So you want to make sure that there is no bioburden at this stage. And you're going to check for like any fungal contamination or the other growth contamination. None of those things should be there. Usually you do not see any of those things, but there are some rare instances where you do come across that. Okay, so after that, what are the challenges in this particular unit operation? You're testing, you need to know how to use the filter type, the material of the filter, whether it is a polyether sulfone, whether it is, cellulose filter. Those are some of the challenges that you need to worry about, because sometimes the plasmid DNA can either bind very tightly to one of these membranes or materials, and they may not come out. So you do not want to lose any product at this stage. So you want to make sure that your choice of the material of the filter is the right one. And you want to size the filter correctly. You do not want to have any blocking or fouling, and you want to choose your flow rates, liter per meter hour. That's like a standard thing, LMH. So you want to make sure that your flow rate is appropriate for filtering, and you're making sure that your plasmid is stable at different temperatures. Like I mentioned, you can store your plasmid at like 2 to 8, which is basically a fridge, -20, which is like intermediate freezing temperature, or -80, which is a much more stable condition. So you want to test your product's stability at different temperatures and make sure that it's stable. You need to define those parameters and understand that. So that's one of the challenges. Sterility and closed system operation. You want to make sure that there's no more contamination. You've already invested close to 3 to 4 days in purifying your plasmid through fermentation, lysis, and whatnot. So you do not want to have any contamination at this stage. And lastly, pump operations, I already spoke about it. The container type, like PETG or HDPE or polypropylene, like you need to know what container is suitable for this plasmid and how it's going to be used. And lastly, tubing material and size, like again, go ahead and Google different tubing, different sizes, and get a general awareness of that, like whether it is size 16, size 17, 35, 36. Cole Palmer has a really good chart and everything, so you can always go into those websites and get some more information. And that's about it, I think. So we kind of like covered all that. Some of the relevant topics for, filtration is over here. And that's it. I hope you got some insight into operations and manufacturing. If you have any questions, this is probably a good time for me to take that.
Thank you, Mukunda, for the wonderful lecture. We have some queries, from the participant sides. Hello?
Okay, go for it.
Yeah. How do you effectively maintain the plasmid stability in the culture?
So like I was mentioning, the stability of the plasmid is dependent on the pH of the solution, and all across the unit operations, right from fermentation through lysis, through chrome, kind of want to maintain neutral pH to about pH 8. That's kind of like important. And the other thing is like you do not want to agitate your solutions a lot. You want to have like gentle mixing everywhere. Otherwise, what happens is like you're going to shear your genomic DNA and you're going to like fragment your genomic DNA, and then it'll get mixed with your plasmid. And from what I've experienced, it's kind of like impossible. Like once your genomic DNA is sheared, you can't separate your plasmid and your genomic DNA because they're pretty much the same size. So that's how you kind of want to treat, be gentle with your solutions, and that's how you keep your plasmids stable.
The next query is, what is the real solution to the problem of inclusion bodies?
That's a sciencey question, and I do not know the answer to that. You probably need to ask some of your research scientists that. A lot of those things I do not deal with on a day-to-day basis, so I'm sorry, I do not know that.
Okay. And then what is the sterilization method you will be using while transferring the inoculum?
So you do not want to sterilize your inoculum, right? Because that's the bugs that you want to transfer. Those are the bugs that are going to like replicate and produce the plasmid. You want to do a sterile transfer, you're not sterilizing anything over there at the point in time. Your inoculum is just being transferred from container A into the fermenter very carefully in a closed system.
Okay. And then which anion exchanger is used?
Sorry, can you repeat that? Which anion exchanger is what?
What is the type of anion exchanger is being used?
So I have, in my experience, I've used like resins like Q-Hyper-D. That's from Paul. Recently Sartorius took it over. And then I've also used, I'm kind of blanking on this, but I can get back to you on that. Q-Hyper-D, we have also used like DEAE, TMAE. I'm forgetting the full forms of it, which is why I'm kind of like blanking, but these are the different types of anion exchange resins that you can use for plasmid purification.
Okay. And then bacteria are prone for mutations. How the integrity of mother culture genome of plasmid is maintained?
So that is dependent on the sequence and the conditions that you provide to the bacteria during fermentation. Let's say you're growing it at, let's say the bacteria is really happy to grow at 37C, but you do grow the bacteria at 40C, then the bacteria may be more prone to mutations. But if you provide the appropriate conditions, like the right temperature, the right pH, the right oxygen levels, then the bacteria need not adapt to anything else. And at that point in time, its machinery is more focused on just replicating itself and producing the plasmid. At that stage, or in that kind of environment, it will be less prone to make mutations.
So how much?
And you check for these things at each stage by doing sequencing. So you will be able to know if there are like point mutations or other kind of mutation.
And the next query is, how much amount of plasmid DNA could be produced at industrial scale?
Oh, these days you can do all the way up to like 100 grams. There is a company called Nature Technology here in Alabama. They have a really robust fed batch process, and they have a really good nano plasmid platform using which they can produce 100 grams. And you might think that 100 grams of plasmid is like a really tiny amount, but when you come to like the plasmid scale, that's humongous. That's a really big number. So 100 grams in about 2 days is like a lot of plasmid.
And the next is, how do you store the large amount of DNA produced at industrial level?
So think about it this way. Even if you're producing 100 grams of plasmid at 1 meg per mil, all you're having is 10 liters. To store 10 liters is not a big challenge. All you're doing it is storing in those 1 liter bottles or 2 liter bottles. To give you a comparison, the Coke or Pepsi bottles that you drink is about 500 ml bottles. So 20 of those bottles, not a big challenge to store in any freezer, right?
Yeah, the next query is whether agarose gel electrophoresis or capillary electrophoresis is used in the downstream processing of plasmid.
You use neither of those, right? That's more of a quality control test that you do, and you use both age and CE to test the quality of your plasmids. Once you do your restriction analysis, you can do either agarose gel or CE to determine the different fragments of your plasmid. You use anion exchange or reverse phase chromatography or Engg to do the downstream operations at the scales that I'm talking about.
So which method is more advantageous for mass production according to you? It's batch, fed batch or continuous?
Fed batch for sure, because you're able to get 3 to 4 times the amount of plasmid that you get from batch processes. Perfusion processes, I've not really come across as much. It's really used more with mammalian cells like CHO and in the production of monoclonal antibodies. That's a completely different organism set. For fermentation, for E. coli, I think fed batch is your best bet.
Okay. So now I request our HOD, Dr. B.K. Manjunath, to speak few words about this lecture.
Good morning, all participants. Hope you people had a very nice time with Mokon. Mokon, it's very amazing. I really felt very good while hearing you in presentation, and you took us into a virtual visit to your industry. Really, it was very nice and brainstorming, informative, and very interesting lecture that I had. And thank you for accepting our invitation and coming back to the department, though virtually. And any other guidelines that you would like to give to our students?
Sure. So, and just to give you an example, I think I had a really good project experience under Manjunath sir. During my 7th semester and 8th semester, we did a lot of paper presentation, poster presentation. We went and presented a paper at IISC, I think, and then we got a good funding from National Innovation Foundation back in the day. It kind of was really good exposure. All I want to say is all of you students will one way or other complete your engineering. There is no doubt about that. But during the 4 years, go and do extracurricular things, go to a different poster presentation or a seminar or a symposium. Those are the things that is going to stay with you for a longer time. Go do some industrial visits. My time, I went to CFTRI, MVIT college had a good program one of the semesters, in between one of the semesters, that was a good exposure for me. Then we went to Sartorius during one of my semester, 7th semester or something. We saw how the fermenters were being made in person. That was like a good experience. And then we went to like PESIT or something for like one of the poster presentations. So those are the memories that I think will be with you for a longer time. And focus on those topics and do learn those. There's a lot more things just other than research, like there's operations, there's manufacturing, there's supply chain, there's like bioinformatics, there is like a whole spectrum of things. So it's good that you've chosen biotechnology. You'll be able to make a difference, in others' lives. So you're on the right track. That's what I'd like to say to the students.
And Mukunda, we have one more question. What about the purity concentration level of plasmid DNA?
So the plasmid DNA is pretty pure, like I would say at 100%. You're able to remove residual protein, residual DNA, host cell genomic DNA to a very large extent. At the very end of it, your residual protein is what in like picograms or like femtograms kind of level. Your antibiotic is also like in like really low nanograms kind of level when you're comparing with your plasmid DNA, which is in grams. So all of those things are like in parts per million kind of thing. So that basically says that your plasmid is very pure.
Okay. So that's all with the queries. So on behalf of the management, staff and students of the Department of Biotechnology, the Oxford College of Engineering, I would like to extend our heartfelt gratitude to Mr. Mukunda, our alumni, for giving excellent lecture and taking us deep into the industrial oriented towards the plasmid isolation. So thank you so much, Mr. Mukunda, for this wonderful lecture. I would also like to extend my heartfelt gratitude to our chairman, Shri SNVL Narasimharaju Garu, for his continuous support. And I would like to extend our sincere gratitude to our principal, Dr. Aravind A.S., and our director, Dr. Amarnath K., for their support and encouragement. And I would like to extend our heartfelt gratitude to our HOD, Dr. B.K. Manjunath, for his motivation, guidance and support in organizing this event. And I would also like to extend my sincere gratitude to all my dear colleagues for their support in organizing this event. A special mention to our student Rohit for his support. And last but not the least, I would like to extend our sincere and heartfelt gratitude to all the participants for attending this 5 days technical symposium on an insight into biotechnological applications. So from on the behalf of the staff, students and management of the Department of Biotechnology, the Oxford College of Engineering, we extend a very heartfelt gratitude to all the participants who had actively participated in our 5 days technical symposium. Thank you all.
Thank you, Mukund, once again, and